and I'm an undergrad at UK. So we are going to be doing a pretty interesting lab today. The main topic is about deep tissue injury. So take for example a construction worker is working and a cinder block falls on his leg. So what happens there is when the block falls, the tissue is pressed up against the bone, and then there's going to be a lot of damaged tissue underneath the skin. So initially there's going to be a lot of cells that die, and these cells are going to affect the healthy neighboring cells and tissue around it. So what happens there? What's the process, I guess? So inside of a healthy cells, there is a high concentration of potassium, amino acids, and a lot of other cellular constituents. So when the cell dies, these are all, it lyses and it all breaks apart. So this high potassium and cellular contents now affects other cells, the healthy cells around it. So when you have a healthy cell that's damaged, what happens is that the cell lyses and releases all this intercellular components, such as high potassium, amino acids, and other intercellular constituents. So this then can affect healthy neighboring tissue. So it's known that this high potassium that's released from the damaged cell can sometimes depolarize or affect the healthy tissue that's neighboring this damaged tissue. So this high potassium not only affects the muscle, but it can also affect the nerve that goes past this damaged muscle. So these effects we're going to investigate in this lab. In this lab, we're going to mimic deep tissue injury in crayfish. Many student labs use crayfish because it's easily viable and you can easily maintain it during experimentation. So how are we going to mimic deep tissue injury in this crayfish? We're going to use a solution of potassium, and we're going to put it on the prep, and we're going to see how that affects the muscle. Second, we're going to use this thing that we like to call muscle juice. So that's homogenized muscle, crayfish muscle, and we're going to put that solution on the prep and see how it affects the muscle as well. So how are we going to take all these measurements? There are 3 different parts we're going to do. First, we're going to measure the resting membrane potential of the muscle fibers. Second, we're going to look at the synaptic transmission of the phasic and the tonic muscle. Third, we're going to look at how the neural circuit is influenced. So let's get started. Okay, so to start off this lab, we're going to have some crayfish saline, and we're going to want to put that on ice to keep it cool. And this is ground up muscle that we're going to use to make the muscle juice later. So we're going to take this crayfish in here, and we're going to put him in this ice, and we're going to put more ice on him to anesthetize him. So we're going to want to pick him up, and you're going to want to be careful when you do this. So you look here. So we're going to want to place him in this ice right here. We're going to hold him down so he doesn't squirm. We're going to just put a good amount of ice on him, and we're going to leave him in here for about 10 minutes, and that will do the trick. So after 10 minutes, we are going to quickly decapitate his head. And now we're going to clip these big claws so they won't hurt you. So now we're going to separate the abdomen from the thorax, and you can see that he's a male from the large stylets. And we're going to try to cut as close to the thorax as possible. So first we're going to have to cut the stylets off. So be careful and use smaller scissors for this. If you can see this line right here, this is where the cuticle meets the ribs, and this is where we're going to want to cut. And make sure to point the scissors away from the back so you don't damage the muscle that we need. So now we're going to cut along that same line on the other side. So what we're going to do now, we're going to pull this up and we're going to cut right along here. And interesting fact, this is actually the GI tract, if you didn't know. So now we're going to take this prep that we've dissected and put it on a sill guard dish. And we are going to cover it with the saline from before. So now we're going to move it to the microscope. This microscope actually has a boom stand, which kind of helps you move around a little bit. And here's the focus knob, here's the zoom, and this is just like eyepieces to adjust to your own eyes. So what we're going to do first is we're going to pin it down along the midline, and now we are going to come in with the scalpel and cut this part right there. So now I'm cutting the midline. So once I reach the end here, we can separate these two parts right here. So now that we've cut that off, we can get rid of this dead tissue. And then we can separate these two, so we can use one half for another dissection or preparation. Didn't cut this all the way. There. You can take this half and put it in some saline. So now what we're going to do is we're going to stretch this out. So this is going to take some kind of a U shape, kind of like this. And so because we've stretched it out and the muscles are nice and tight, when we stimulate it, the muscles aren't going to want to pop out the intracellular electrode. Right now we're going to make the electrode, and we're going to use this multi-pipette puller and this glass that we bought. What's going to happen is we're going to slide this in right here, and this coil is going to heat up, and it has a pulling mechanism which is going to pull it back, and it's going to make a really nice sharp tip that's about a micron. So we're going to put this in, gently put it in through the coil and into here. Oh. Then we're going to tighten this a little bit, not too tight, to make sure it doesn't break. going to tighten this as well. We're going to push this all the way to the front. We're going to tighten this. And we can put this in here and close it up. Turn it on. And your instructor will help you with this part. It could be pull 1 or pull 2. So this is the final micro electrode. You can see that it's pretty sharp at the very end. So now we're going to put the 3 molar potassium chloride, and the important thing is to not let the potassium chloride drip over the end, so it won't get in the saline and affect our experiment. So we're going to want to tilt it like this so it can drip from the back. I'm going to slowly fill this up with KCl. So sometimes you're going to get some bubbles, so you can kind of shake it around a little bit. So one more thing we need to put in there is our ground wire, and you'll have like a little piece of clay to stick it on there so it won't move around too much. So now we're going to test the resistance of the electrode. So you're going to want to carefully put the electrode in the saline and make sure you don't break the tip. So now we're going to go over here to the amplifier. We're going to turn it on, and we're going to press the probe. And if it's not already at 0, you're going to want to fiddle around with this DC offset to make it 0. And now we're going to press the electric test. And so you want, this is perfect. You want it to be around 20 to 60 megohms. So this fits in that range. So it has a good enough resistance to continue the experiment. So sometimes you might have to ground the amplifier to the Faraday cage to cut out the 60 cycle. You also might have to unplug your light source to take out some of the noise. So now we can go over to the computer and start the lab chart. So now to get our data on the computer, we're going to hook up the amplifier to the analog digital board. So you're going to want to hook it up to the 10X right here, and then hook it up to input 3 in the power lab right here. And then we're going to go over to the computer, going to X out of this, going to go to setup, channel settings. And we only want 3 channels. So we're gonna change this to 3, and we don't want the first 2. And just do that and just check channel 3 and say okay. And because we don't have the first 2 channels, we can just take this and drag it up so channel 3 is on the entire screen. So then you can just hit start, and you can see all of this, right? And that's because we still have our electrode test on. So if we turn that off, then you could see the baseline. So what you're gonna first wanna do is you're gonna wanna put this clay, wax, whichever one, next to your dish so it won't move around when you're poking it with your electrode. And you can use the knobs, the coarse knobs here, to move your electrode around gently. And so you can figure out how to get the tip of your electrode right over the muscle that you want to stab. So you can either start with the phasic muscles, which are the L1 and L2, or the tonic muscles, which is the SCL. So it's going to be kind of difficult to see the electrode, so you're definitely going to have to use this microscope. And you're going to want to push it in to the muscle a little bit, and when it goes into the muscle, you're going to see it dimple just a little bit, and then if you pull out, you can see it like the muscle moving with the electrode a little bit as well. So that's how you know that you're inside of the muscle. And you can also kind of tell through your reading right here. So I have a reading that's about -78, 77 millivolts. So I know I'm in the muscle right now. And now we're going to pull it out, and we're going to do it a couple more times just to get multiple readings. So what you're going to want to do is, after you got the first one, you're going to want to move your electrode over just a hair so you can get another reading on another muscle fiber, and you're going to see a dimple just like before. And so after you got your 3 readings, you can average it to find the potential. So after you got your 3 readings, you're going to want to carefully back out the electrode from the muscle fiber, and then you're going to want to hold on to the manipulator and gently loosen this so you can pull it out. If you go too quickly, the electrode's going to break in the solution, and that would be a problem. So then you're going to want to take this ground out and take out the clay. And then we're going to want to dump this saline out. And you're going to want to put either the potassium solution or the muscle juice. So you're going to want to be really careful not to hit the electrode with your hand or the dish or anything like that, because it could easily break. And don't forget to put the ground back in, because that's also important. And after you did that, we can stab the muscle again 3 times and then wash it out with another solution and stab it again, and keep doing that until you have all of your trials in.